old-fashioned approach at this point because we already went through the PowerPoints I have posted for you. A few issues might come up on your exam that we haven't covered because they actually are stressed in a different chapter. Like I said, I don't like the way this book is organized, but I'll give you a heads up if it is. And here's one I did mention, but I want to be very clear about this. Okay, gram-negative, gram-positive bacteria. You need to know the difference. Not only in terms of the thickness of the wall, the result of the gram stain, but you also need to know, like penicillin works on gram-positives, not on gram-negatives, because we did that in lab. And there was something else I mentioned. Remember gram-positive cell wall, 6 to 8 layers? I'm not going to have enough room. Let me move it over here. Oh, can't write today. 6 to 8 layers peptidoglycan with, do you remember the teichoic acids? I didn't post chapter 4 PowerPoint, so you need to go to chapter 4 and look at the picture of peptidoglycan. In fact, does somebody have their textbook out?
Yeah, I do.
Yeah, good girl. Find chapter 4, the structure of peptidoglycan. Trying to rush. That doesn't work.
Let's see. Chapter 4. I'm not sure if it's chapter 4. Are you sure it's chapter 4?
Isn't it structures?
No, it's microscopy standing on.
Oh, it's chapter 3 then. See, in my old book it was chapter 4. I still remember that. Cell structure and function. Where's the peptidoglycan?
Somewhere.
Is this plasma membrane? Yeah. You'll find peptidoglycan in there. I hate the way this book is organized. Plasma membrane.
Cell wall.
There you go.
Bacteria
Here.
cell wall here. This one.
Yeah. So this is page 64 and 65. All right. Now, we wrote on the board general differences between gram-negative and gram-positive cell wall, but I want to emphasize this because I only mentioned it briefly. Gram-negative bacteria have something gram-positive bacteria don't have, this periplasmic space. The periplasmic space is a space between the cell wall and the plasma membrane. It's a protective space that only gram-negative bacteria have. And it protects it from even its own toxins in its cell wall, but also from other chemicals and toxins that would try to enter the cell. It sequesters them away in that space. So this is something unique to gram-negative bacteria. Gram-positive bacteria don't have a very complex wall. It's just 6 to 8 layers of peptidoglycan. But what they have in it are teichoic acids and lipoteichoic acids. And they show you this in the figure on page 65. So this is something unique to gram-positive bacteria. This is something unique to gram-negative bacteria. Okay? Reason I mention it is because, like I said, sometimes I pull test bank questions from the textbook author, and he does have a couple questions about that. Remember, your gram-negative bacteria have only 1 to 2 layers of peptidoglycan, but they have that outer membrane over it, which consists of lipoproteins, phospholipids, lipopolysaccharides. The major one to remember is your endotoxin, lipid A. It's a lipopolysaccharide. And they'll ask you about that if you get that question. Also, we didn't talk a lot about it, but if you look at the foodborne, 5 big foodborne illnesses, it's posted, one of the posted things in Canvas, and it will show you a copy of this. This is what they hang in like restaurant kitchens and stuff for the employees. The big five foodborne illnesses: hepatitis A, that's a virus. Norovirus, that's a virus. Not worried about those for this test yet. But here's 3 bacterial: E. coli, and not just any ordinary E. coli, Shiga toxin producing E. coli. Shiga toxin is a really severe toxin, going to give you cramping and really bad diarrhea, but could also go into that hemolytic uremic syndrome. So it's called Shiga toxin because Shigella also can produce a Shiga toxin. So it's a Shigella-like toxin. It's a really bad toxin. These 2 bacteria can produce it. And also we have Shigella and we have Salmonella. Here, Salmonella typhi is typhoid fever. So it could also be Salmonella enteritis, which is very similar. It's just not typhoid fever, but it is a bad gastroenteritis. Okay. All right, so let's see. We didn't talk about antibiotics yet, but I did write a little chart here that talked about bacterial structures and what type of antibiotic targets different bacterial structures. Do you recall what antibiotic targets the peptidoglycan of the cell wall?
Penicillin.
Penicillin, good. And it gets gram-positives, much better, doesn't usually get gram-negatives. And that's because gram-positives have a lot of peptidoglycan and that's all they have. So if penicillin can disrupt the peptidoglycan, the gram-positives are really going to be affected. The gram-negatives aren't affected as much. They don't have much peptidoglycan, so it's not a big target. And they have the outer membrane to protect them from the penicillin. And even the periplasmic space might help protect them from the penicillin. You might not remember this, but in your groups of Streptococcus, Enterococcus is what used to be called Group D Streptococcus, and it's naturally resistant to penicillin. It's naturally resistant to penicillin, Enterococcus is. You know Staphylococcus and Streptococcus? What penicillin's going after. But Enterococcus is naturally resistant to penicillin. Guess what else is naturally resistant to penicillin? Most of the gram-negatives, like E. coli. Couldn't get E. coli with penicillin because it's gram-negative. We also talked about plasma membrane. Actually, I wrote it on the board. Plasma membrane. What disrupts the plasma membrane? Talked about polymyxins. Stuff like neosporin that you put on your boo-boos. That targets the site of the plasma membrane. Also, we talked about ribosomes. What do ribosomes do for a cell?
Protein synthesis.
Right, protein synthesis. So tetracycline targets ribosomes and inhibits the cell's ability to synthesize proteins. So a bacterial cell can't make its proteins when you hit it with tetracycline. So those are probably the most common ones, but go back and review that little chart that I put on the board for you. All right. Actually, that's just a little preview of antibiotics and how they target bacterial structures. When we do chapter 10, that's the chapter on antibiotics, and we'll work on that mostly in lab. So you'll get that information in more detailed form then. But for now, you need to know the structures and which antibiotics target them. And if it targets them, what's going to happen? So if it targets the cell wall and you can't make that transverse plate in time for cell division, the bacteria is going to spill apart. ribosomes inhibit protein synthesis if you hit them with tetracycline. Also, you need to know your different bacterial structures, what they do. And you have to compare them to eukaryotic structures. So remember that table we put on the board, differences between prokaryotes and eukaryotes? Prokaryotes have only one membrane. What is it? What's the membrane in a bacterial cell? The plasma membrane. It's the only membrane they have. Eukaryotic cell has a plasma membrane plus other membranous organelles. Like, for example, what organelle that's made of membrane do eukaryotes use for making ATP?
Mitochondria.
Mitochondria. Bacteria don't have mitochondria, so what are they going to use for making
Plasma
ATP?
membrane.
Plasma membrane, good. And you also have to remember structures like for bacteria. Like, for example, eukaryotes can have cilia on the outside of some cells. How about bacteria? Can they have cilia? No. But bacteria can have flagella, like eukaryotic cells can have flagella. Not too many eukaryotic cells have flagella. In fact, in kingdom Animalia, only one, the sperm cell, has flagella. But for bacteria, a lot of bacteria have flagella, but never cilia. Do you remember what a pilus is? And do you remember the plural for pilus is pili or pili? People would actually say pili, I say pili. Remember what pilus is for? Conjugation, right? Conjugation is exchange of a plasmid between bacterial cells. Remember what a plasmid is? It's an extra set of genes, like 5 to 10 extra genes.